world why our immune system starts to attack the very body where it resides. Today we are talking about the 2025 Nobel Prize in Physiology on medicine and how 3 scientists figured out how our immune system knows when to stop attacking. This discovery is changing how we treat cancer, autoimmune diseases, and even organ transplants. Stick around because by the end of this video, you'll know what regulatory T cells are, why the gene FOXP3 is a big deal, and how this all connects to real-life medicine. Our immune system is like a high-tech security system for our body. It has to do 2 things at the same time. Attack dangerous invaders like viruses and bacteria, avoid attacking our own healthy cells and organs. When the second part fails, we get autoimmune diseases, things like type 1 diabetes, rheumatoid arthritis, where the body's own defenses turn against itself. For a long time, scientists knew the immune system had ways to avoid attacking the body, but didn't fully understand how it stayed under control. In the 1990s, a Japanese scientist named Shimon Sakaguchi made a breakthrough. He discovered a special type of immune cell called regulatory T cell, or Treg for short. If normal T cells are like security guards that attack intruders, regulatory T cells are like supervisors who tell them, calm down, the job is done. Stop attacking now. Functions of Treg cells include self-tolerance, preventing autoimmunity, avoiding fetal rejection during pregnancy, controlling chronic inflammation, and regulating immune response in infection to calm the immune system. Sakaguchi showed that when these regulatory T cells are missing or not working properly, the immune system goes out of control and starts attacking the body's own tissues. This was the first big clue that there is an active brake system built into our immunity. This is where Mary Brunkow and Fred Ramsdell come in. They studied a strange strain of mice called scurfy mice. These mice developed horrible autoimmune disease and died young. Now the male mice, they developed this fatal autoimmune disease, dying at 3 to 4 weeks old. But the female mice, they do not develop a severe autoimmune disease and stayed alive for longer. This made them hypothesize that the gene responsible for the disease might be present on the X chromosome. Brunkow and Ramsdell searched approximately 150 million DNA base pairs on the X chromosome and pinpointed FOXP3 in 2001. Finding the error was like searching for a typo in an entire library full of words. After a lot of hard work, they finally found the single faulty gene. Then they checked human patients with a rare disease called IPEX, where children developed severe autoimmune problems, and found mutations in the same gene in humans. A couple of years later, Sakaguchi and other researchers showed that FOXP3 is the master switch that creates and controls regulatory T cells. Shimon Sakaguchi's group found that the FOXP3 gene is expressed in the regulatory T cells, and it also happens to be the main gene responsible for formation of regulatory T cells. So now using this, they could create regulatory cells from T cells in a dish. Now Sakaguchi and Brunkow Ramsdell studies further converged by the finding that the scurfy mice who do not have FOXP3 also do not have regulatory T cells. Now that we can make regulatory T cells in a dish, can we treat conditions of an overactive or self-attacking immune system by introducing Tregs? There are over 200 clinical studies working on finding an answer to this very question. Autoimmune diseases are not the only disease conditions that can be potentially cured by Tregs. They are now being tested for curing asthma, inflammatory bowel disease, and skin conditions. Regulatory T cells are also being tested for reducing the risk of rejection of an organ. We know of conditions where the presence of regulatory T cells can cause harm. Tumor is one such example. The tumor cells shield themselves from the immune system by recruiting regulatory T cells. And for treating these tumors, we need to get rid of Tregs. The Nobel Prize went jointly to Mary A. Brunkow, an American scientist who helped discover the FOXP3 gene's role in immune regulation. Fred Ramsdell, an American immunologist who worked with Brunkow on the genetics of immune tolerance. Shimon Sakaguchi, a Japanese immunologist who discovered regulatory T cells and later linked them to FOXP3. Together, their work solved a huge mystery in immunology: how the body avoids destroying itself while still fighting infection. But there are several open questions that remain. What controls FOXP3? How does the thymus and immune system select Treg cells? Are Tregs involved in creating immune tolerance towards our own gut bacteria? So, to sum it up in one line, the 2025 Nobel Prize in Medicine honored the discovery of the body's own immune brake system, regulatory T cells controlled by the FOXP3 gene, which is now guiding new treatments for cancer, autoimmune diseases, and transplant rejection.